Hello and welcome to Nikolay's genetics lessons, and today I have prepared another video about gel electrophoresis common mistakes. And if you will take a look at this gel, you would instantly notice that some of the lanes are absent here, and those lanes which are present and have bands, but you see that bands not clearly visible and we cannot distinguish them well. And there are a number of artifacts present here, like this line that goes across the gel. So what caused all these artifacts, and what's the problem with this gel? It looks like this problem was caused by badly formed wells or careless loading of the sample into the gel. The background, as you see, is dark and uneven, and that suggests that proteins were in the running buffer itself. Most likely apparatus was jarred and much of the samples were slopped out of the wells, and the spilled sample would not resolve into the bands, of course, since it is continuously entered the gel from the upper buffer compartment, and not the presence, as I said before, of the horizontal line here that would suggest that the wells were too shallow and some material spilled into the adjacent wells. This symptom also shows up in perfectly good gels when the electrodes are hooked up backwards for a few minutes. Protein migrates out of the wells instead into the stacking gel. If you would notice this problem within few minutes, you can save some information, but the pH effect that cause efficient stacking is compromised. Your sample would be lost and protein would contaminate running buffer. This is exactly what we see in this gel. So the problem can be resolved very easily. Do not overload wells, and again, as you see, we also see continuous line here. That means that wells here had a leakage and proteins just went across the lines. So this is the problem with this gel. So make sure you not overload your wells, you not spilling material, and that wells of the size that is match your sample that you load. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.